Welcome back, dear scientist-to-be. Today, we're diving into a famous family of lab techniques known as blotting. Get ready to meet the southern, northern, western, and even eastern blots and find out what each one is used for in the lab. And we're back. Blotting techniques are essential molecular biology methods used to detect specific DNA, RNA, or proteins from a complex mixture. But why the strange names? Their names reflect a playful naming tradition that started with the Southern blot. The Southern blot is named after its inventor, Dr. Edwin Southern, who developed the DNA detection method in 1975. As a pun on Southern, the Northern blot was developed to detect specific RNA, and the Western blot was created to detect specific proteins. The Eastern blot extends this naming tradition by detecting protein modifications, though it is less commonly used and often considered a variation of the Western blot. So what are their main purposes? Each technique targets a different type of biomolecule and uses a distinct detection strategy. Southern blot. This technique detects specific DNA sequences. After separating DNA fragments by gel electrophoresis, they are transferred to a membrane and then detected with a labeled DNA or RNA probe that hybridizes to the target sequence. This is useful for gene mapping, mutation detection, and DNA fingerprinting. Northern blot. This technique detects specific RNA, usually mRNA, using a similar method to the Southern blot. RNA is separated under denaturing conditions to prevent secondary structures, transferred to a membrane, and then detected with a labeled probe. It's a key tool for gene expression analysis. Western blot. This is the most widely used blotting technique in modern labs, especially for protein detection. Proteins are first separated by SDS-PAGE gel electrophoresis, transferred to a membrane, and then detected using specific antibodies. It's highly specific and can identify, quantify, and analyze post-translational modifications of proteins. Eastern blot. This technique is a variation of the Western blot used to detect specific protein modifications such as glycosylation. It uses probes that specifically bind to the modification on the protein. The key difference is the detection agent. Southern and Northern blots use nucleic acid probes, while Western and Eastern blots use antibodies or other specific probes. The choice of which technique to use depends on the research question and what you're trying to find in the sample. Wow! From the original Southern blot to the widespread Western blot, this family of techniques is a perfect example of how foundational tools in molecular biology evolve. Each one is a specialized method that allows us to get a precise look at the specific molecules we're interested in. And speaking of the Western blot, in our next episode, we'll be doing a deep dive into the practical, on-bench protocols for this powerful technique. So if you're as fascinated by these classic lab tools as we are, hit that subscribe button right now. And don't forget to ring the notification bell so you never miss a single revolutionary discovery from the Geneticist. This episode is just one part of our exciting How To Biotech Lab playlist, so dive deeper there for more incredible insights. See you in the next episode, inside the amazing world of the cell.